Hi, my name is Nicole Delrosso. I'm a fifth-year biophysics PhD student at Stanford in the labs of Locker Bintu and Polly Fordyce. I'm fascinated by how the genes in our cells are turned on and off. The proteins that do this are called transcription factors, and there are actually over 1,600 different transcription factors in our cells. There are two independent parts or domains to these proteins, the DNA binding domain that's structured, and the effector domain that's largely unstructured. The DNA binding domain localizes the protein to specific DNA sequences in the genome. And these domains are actually how we identify these proteins. So every transcription factor has an annotated DNA binding domain. But the other part is this effector domain. And it's thought that these domains regulate transcription by binding other transcriptional cofactor proteins. They can activate, repress, or both activate and repress. And those we call bifunctional domains. However, we're missing annotations for more than half of these effector domains. We just don't know which proteins contain them and where they are. Furthermore, even though interactions between effector domains and cofactors are essential for converting protein binding into transcriptional signals, we're currently missing quantitative measurements describing these interactions. So during my PhD, I've been addressing these questions by first using a high throughput method called HT-Recruit, which I've diagrammed in the upper left, where we use next generation sequencing to measure the transcriptional strengths of large protein tiling libraries. You can see an example of what one of these protein tiling maps look like for a single protein, this MIB protein, where activation is in yellow and repression is in blue. And in total, with these measurements, I annotated thousands of effector domains, hundreds of which are novel, which makes our study the largest resource for human effector domains to date. Now, we have this list of hundreds of activation domains, but we still don't precisely understand how these domains are activating gene expression. There are only several coactivators, so how do these few coactivators bind to all the hundreds of biochemically distinct activation domains? It's been a hard problem to answer because these activation domains are intrinsically disordered, as I mentioned before, they're weak binders, and they're difficult to express. So our solution to this has been to turn to miniaturization, where I developed a microfluidic platform that allows for recombinant expression and purification of thousands of effector domains in parallel, followed by direct measurement of cofactor binding affinities. We call this platform stamping, which stands for simultaneous trapping of affinity measurements via a microfluidic protein-protein interaction generator. And to date, we've used stamping to measure binding affinities for over 1,000 activation domain coactivator interactions. So we think that with these measurements and our systematic approach, we're able to provide a really rich resource for understanding the function of human transcription factors, for engineering compact tools for controlling gene expression, and refining predictive models of effector domain function. Thank you for listening, and thank you again to ARCS for all of the support.